different scale, right? We work with insects and many of them. So basically, I study predation. We've just heard about how important it is to study natural guided behavior, and I think that predation is as good as it gets. It's innate and it's robust, so I don't have to teach my animals to do their thing. It's necessary for survival, right? If they don't catch anything, then they die. So it's in a constant arms race. It's pushing the neural system constantly. It's tractable because these animals are small, and it's highly diverse. So we can actually compare what happens when you put different ecological pressures. And I study one behavior, predation, but there are many, many steps to it that are very important from a neuroscience point of view, right? The animal has to detect the target, it has to select it, it has to approach it, right, in order to catch it. So how do they do this, and how do they improve the neural performance so that they get better than their prey? This is kind of what we ask. So if you want to do that in aerial animals, you may want to start with dragonflies or dancerflies. We have an internal arena in Minnesota, where we can actually video this behavior, and you see this is how it works. You can see many species in here. This is a high-speed video of the behavior. It's about 500 milliseconds. And you can see that this dragonfly just took off to catch that target. It's moving independently, the wings. At the right time, it puts out the legs, and it basically catches the target, right? So it's doing all of this within 500 milliseconds. Now, dragonflies are thought to be very, very ancient insects, which makes them very interesting from a neuroscience point of view. But what people actually forget is that there is a sister group, the dancerflies, and that actually provides for really neat comparisons, because most dragonflies have basically merged the eyes at the top, so they make them holoptic, whereas all the dancerflies have kept them really far apart. So we have a system here that within about 300 million years ago, they had an ancestor that was already predatory. And these two animals are predatory, but they've obviously done something very different with the sensors. So we wanted to know why and what that means. So you can see here in the field, we also go to the field to record what these animals do, not just in the lab. And you will see here a few sequences of dancerflies. These are dual wings catching targets. And what you can see is that instead of going upwards like you saw the dragonfly, they basically go forward, right? And it's a very fluttery type of flight. It looks different already. So we analyzed this in 3D, and indeed what we found is that dragonflies basically sample the targets with the dorsal fovea. We already knew this, whereas dancerflies sample the targets with the frontal fovea. And they attack frontally versus dragonflies attack dorsally. We wondered whether they were actually using the same system or a completely different system, and it turns out that the premotor neurons that take the information from the brain to the wind motor centers is normally about 16 of them. They're homologous between both of them, so we record intracellularly from all of them, right? So the animals are basically doing this different behavior with a system that is homologous. So what has changed between them then? And then Jack in the lab did recordings from the dancerfly, from dragonflies, and from these neurons, and what he found, and this was already new from 2012, is that in dragonflies, when you record from these neurons, these neurons care about half of the receptive field, right? They literally have merged their eyes together and made this midline between both eyes. And if the target is basically, the image is placed here at the midline, if the target moves to the left or to the right, one neuron, and only one neuron basically picks that up, depending on which way they're going. With the dancerflies, it looked very different. He will get like the spike trigger average, will get this blob in the middle, right? And we're like, okay, where is that coming from? Is it coming from both eyes? Are they both just as important? So what he did was a classic experiment, right, of just patching the eyes and see what happens to these receptive fields. And he found neurons that we call binocular only, where you need to have the input from both eyes to get basically any spikes at all, right? Then you got neurons that still responded in half of the receptive field. If you patch, one eye, then you get the other half is still there. If you patch this one, you still get the other one there. And I should mention the number of spikes always goes down. So all of these cells are all heavily binocular. It's just that in some of them, everything goes, but in some of them, you see some spikes still getting through. And then the third type, which is that if you patch one eye, then nothing gets through, but if you patch the other, you can get some spikes through, right? So what we think is that this can actually be very easily modeled if you think about summation through threshold, right? There is inputs coming from both eyes, and basically in these neurons here, the threshold is so high that none of the inputs are getting on their own over it, so you don't see any spikes. Here, the threshold is maybe lower, maybe the animal is hungry. We don't know what actually changes the threshold. This will be important to know for future studies. And so on their own, each of the information from each of the eyes gets over threshold. And then in this model here, maybe one where the input from both eyes is actually not balanced. it's weighted, right? So one of them gets over threshold, but the other one doesn't. And so we're really interested in understanding whether this is a property of the neurons. Intrinsic, there's only 16 of these neurons, right? So is this something that the animals learn through experience? Is it tunable, or is it just fixed? And so we'll be doing that next. But if you wanted to know more about that study, then I encourage you to go and look at this new publication. And Jack did an amazing work at putting all of this together. It's a huge amount of work to get out in the field, to be in the lab, to do intracellular, to do spec sorting, all of it together, and in 2 different species. Okay, but I love dragonflies and I love dancerflies, but they have a problem. We have a problem, right? Or a challenge, I should say, which is that the closest model animal is a fruit fly, right? So if we want to take the framework about how the insect brain works from a fruit fly, and we want to take the information that we learn about these animals and kind of match them together, it's kind of impossible, right? For many reasons. One of them is size, right? This brain is much, much, much bigger, has many more neurons. But another one is also just morphology. Not only this animal has 4 wings instead of 2, which matters with how you fly, but the mushroom bodies of dragonflies don't have calyxes, for example. The lobulas have many, many, many more layers than in fruit flies. So, and sometimes you're in the middle of this brain and you don't even know where you are. We don't even have an atlas for this brain yet. So what I thought when I was doing my PhD is that why don't we actually look at predation of dipterans, animals that are much, much closer to fruit flies, but that also do predation, and then we'll be able to compare things much, much easier. So in my lab at the moment, we study 4 predatory diptera flies, so they're true flies, right? So they only have 2 wings than the halteres. It makes it much easier to compare with Drosophila. We study them, very different ones, because they have different sizes, right? So this one is 4 millimeters long, so it's very, very close in size to a fruit fly. And this one is huge, right? All of these 3 are rubber flies, so they're very closely related. If you're gonna fall asleep this morning, so just take this message home, is that although all of these 3 animals are rubber flies, this one, the data that we have at the moment tells us that it uses motion parallax to basically select the prey, detect and select the prey. That one, as far as we understand on everything that we've done, is using stereopsis, which is incredible for a fly, hasn't been shown before, and this is what the data is telling us. This one is a beetle hunter, it only hunts beetles, and we were actually wondering how. And it turns out that it actually samples the wing beat frequency of the beetles, and that's how it tells them apart. And then it does something else, right? And this tiny, tiny one that has, very bad eyesight, it basically uses angular size and angular velocity to kind of proxy for what it will select. So I'm making this point to say I think it's important not just to study a naturalistic behavior, but to understand what the animals are meant to be doing. So if you want to understand the brain, you have to know what they have evolved to do. So I'm going to just focus on these 2 today. They are the smallest, and I think that actually provides for a very, very good opportunity to understand what happens at that scale. If we start with the killer fly here, this is what they do. Killer flies are basically built and designed to be nimble, right? So you'll see a white fly flying out here, and this is again a high-speed video recording. And killer flies are sit-and-wait predators, so they will actually take off and hunt down, fruit flies, white flies. And what you will see is that it misses on the first go, but it turns around really quickly, right? And then it has another go, and then again, right? So killer flies see very poorly, but they're really, really, really fast. Actually, they have the fastest photoreceptors that have ever been recorded. And being so small helps them in being so nimble, right? So that's the strategy here. If you compare it to this Hococephala fusca, you can see the eyes are actually wider than the body, right? It's forward-looking. It has this fovea down here in the middle, and we were wondering how well this animal can see. And so some of you may not be able to see this detail, but I'll follow with the pointer. This is a high-speed video. This is a fly that just took off here, one of these flies. We have a bead all the way up there, and we were trying to figure out if we will see it and hunt it, right? And so you can see the flicker of the fly going all the way up there. Sam starts to react now. You can see he's very surprised. And this fly goes all the way up and catches this fly, this bead, right? So Sam was really, really surprised. You can see the time frame. I think this is great to show our reaction time versus this fly's reaction times, right? And so I think this is important because it shows us that this tiny, tiny fly can get a special resolution in the eyes that matches the ones of the large dragonflies with the really, really big eyes, right? So it's basically cheating the system and putting a huge fovea down in the middle and matching what this animal can do. The body size and the head size is very similar to the killer fly that I've shown you before, but the killer fly is in a spatial resolution an order of magnitude worse off. And this is already 3 times better than what a fruit fly can do, right? So that was in the sensory layer. What about the premotor layer? How are these neurons comparing for these 2 flies? So if you look at the ventral nerve cord of a fruit fly, the only large axons that you see are those of the giant descending neurons, right? Because the animal needs to escape really fast. You make your axons really large. If you look at these predatory animals, what you see is all these really, really large axons here, which we actually thought maybe the equivalent of the target selected descending neurons that I just talked about in Oronata. Maybe it's something equivalent then in these flies as well, because the information about a target has to get from the brain to the thoracic centers. And indeed, when we record from the ventral nerve cord of these 2 flies, and you can do this, we do this in the field, so we ship everything for this animal, for example, we ship everything to Pennsylvania, and we set up everything there. And this is one of such recordings. That's 2 millivolts of extracellular spikes. So because they're so big, right, they give you a really big signal. And so this is just the rig with the tiny, tiny fly here, right? And we're recording from the ventral nerve cord, and we're just moving targets. And so these neurons respond to the moving targets and only to moving targets. And each of them like kind of a different direction and such. But what's important for this data, when we compare between the 2 animals, remember we want to know what's the difference between the 2 of them. What's the latency? So the robber fly, the one that hunts this, long distance and has really, really good acuity, has a latency of between 15 and 20 milliseconds, okay, between presenting the stimulus and it being in the descending neurons, which is really good already, 20 milliseconds delay. However, we were very shocked to find that killer flies, the latency between presenting a visual stimulus and us recording it in the ventral nerve cord is between, and this is work that Jack did, by the way. He's a PhD student in the lab who just graduated. That latency is between 5 and 8 milliseconds, right? This is crazy. This has to be going down directly from the optic lobes through gag junctions. This is how pushed these animals are for speed. So then what have I told you so far about the comparison between these 2 animals? Is that this robber fly has basically the best spatial resolution of any insect, right? Whereas this one has the best temporal resolution that has ever been recorded. So you can see how they're being pushed to very, very different adaptations. This one, we think it actually does absolute prey detection, because you have to understand once you are going to fly for such a long way, you want to make sure that you're hunting the right thing, right? And it makes sense that it's using absolute size to select the prey. This one actually sees very poorly. It just basically uses heuristic rules of prey angular size and angular velocity. If you match that, you can get them to take off after anything, big beats, doesn't matter, they will take off again and again and again. This guy, they do long distance attacks, right? 85 centimeters, which is a long way for a fly that is only 6 millimeters. Okay? These guys do short distance. The maximum that they do is 12 centimeters because they can't see any further than that. They just can't see the prey, right? The sensors don't allow them to do that. They have a relatively long sensory motor delay. These ones have a very short one, right? So you can actually see how the system is consistently from the sensors to the premotor tuned to exactly the behavior that they're doing and the environment in which they're doing this. This animal foveates the target. When it sees something, it looks and it tracks it. It makes sense. If you have a phobia, you have to do that. But you have to take into account the direction of your head compared to your body. This one doesn't. It has equal resolution all across the eye, so it doesn't care. So when it sees something, it just basically takes off, right? So again, it primes velocity and speed of the reaction. This one hunts against a clear sky. Okay, so you can actually have time to look at the prey and decide what to do and then intercept it. This one here, as you saw before, hunts in all directions and in very cluttered environment. So it's very, very different challenges. So then given all of these, what Sam, who did a PhD in my lab, asked was, do they both intercept the targets using the same strategy, or have they actually come up with something very different? And we know they both intercept the target. We can see that from the 3D reconstruction of the videos. Okay. What he first found is that these animals seem to be doing parallel navigation, right? So if you do a line of sight between the predator and the prey at different points during the behavior, it looks parallel. So you go, okay, under these circumstances, the flies could do something very simple to say you just have to close range and you can just keep this line as parallel as possible, right? And this is basically what, and then eventually you will get to the target, even though you don't know anything about the real velocity of the target or the distance to it. And you can do this with a simple feedback controller, such as ProNav, right? You just need to know the gain, the constant, right, that you're going to apply, and you're going to rotate proportionally to the rate of the line of sight. So if this animal turns or changes velocity, that line of sight is going to change, and you're going to use the rate of that to 10 proportionally to it. So Sam basically digitized a huge amount of trajectories, hundreds in the case of the Holcocephala, and then he said, okay, what's the rotation of the line of sight, and what's the rotation of the heading of the predator? Do they correlate? And if they do correlate, what's the gain, what's the constant that I have to give to it for it to actually match? And he found when he did that for Holcocephala, the gain is 3. And this matches very well like an optimal gain that you will do. That's what people do for missiles when they do ProgNav, for example. What was really interesting is that when he did the same for Coynosia, for the skillerfly, the gain was 1.2, much, much, much lower. He could find that fit, but much lower, right? And he also found, again, and this fits the electrophysiology, is that the mean delay that he needed to use for all this to fit really well, for the roverfly was 28 milliseconds, but for the killerflies was 18 milliseconds, right? So the behavioral analysis is basically telling us, is matching the electrophysiology that we have recorded from these animals. And so here is just basically a model in orange, and what the fly did in true life was in red, and you can see that they match pretty well even when the target is erratic. And we think that this matches really well because these animals are very small. They don't have to worry so much about things such inertia, for example. So it simplifies the problem hugely. When we try and do this with the big flies that I talked to you about, then things change completely because now those flies have to worry themselves about getting themselves up to speed. When they do a bank turn, they can't just do it quickly. That takes time, and then that takes energy. And so I think it makes a lot of sense to study animals at different scales and see what applies. Okay, so these are the results of the fits when you do an N of 3 or 4 or such, for Holcocephala, and this is when you do an N of 1.5, 1.6 and such for killer flies. So it fit really, really well. And then Sam did one last thing, which is really neat, which is to say if you take a killer fly and you feed it with the delay and the gain of killer flies, then things work out really well, right? But then you take a killer flight and then model it with the delay of the roverfly and the gain of the roverfly and see if it actually makes any difference. And what happens in such circumstances is that the fly goes completely out of control. It oscillates out of control, right? The gain is too high and the delay is too long for hunting at short range. So I think this is a really good example of understanding what the flies are doing and how something is optimal, because any engineer will tell you that a gain of 1.2 it's not optimal, that it should be 3. But under these conditions in which these flies are working, that is optimal for what they're doing for short range clutter environments. Okay, and this fly actually behaves a lot more like a missile. Okay, so just to finish, I just want to say that in my lab we are very much driven by understanding what the animals are doing. I collaborated with Trevor Wodiel, who is now famous for putting glasses on cuttlefish, to understand the behavior, the attack behavior, the predatory behavior of cuttlefish, and what cues are they using, because we want to know that first, right? What cues are they using to make the decision to attack? These are not the only animals that we are putting glasses on. So in collaboration with Jennifer Talley, we are actually putting glasses on these large roverflies, and they do the behavior with the glasses on, so that opens up a huge amount of, really, really exciting research for us. So just to reiterate that we want to understand the how and the why of brains, and I think very, very, very first thing that we need to understand is what those brains evolved to do, so we test them for the right thing. Okay, so with that, to thank the 2 PhD students that did most of this work, Sam Fabian and Jack Sapple, and of course the collaborators Trevor, Karen, Jennifer, Rob, and Han Xuan, who always keep me on track. And we're doing, these flies will actually do that head tracking while you hold them down, while you put glasses on them. So we have a fantastic system now to understand what happens when they track targets and such. Okay, thank you.
Beautiful talk. Now that you have this exciting behavioral information on different species, what's your strategy to get inside the circuit, like on the comparative study of the visual system itself? I don't know, comparing like difference in the number of layers or like proportion of cells.
So we have a grant at the moment to do this, to get in between the sensory and the motor, right? That's a goal. The goal is to basically develop two-pronged approach. One is to develop genetic tools, right? I always kid around that I do have genetic tools, it's called evolution, but we do have to get genetic tools. We have the killer fly colony. We can breed killer flies in the lab in really big numbers. The only challenge at the moment is that the killer fly larvae are also predatory. The majority of these flies, so that's a whole really cool behavior to study, but it makes it challenging for creating genetic models. But we are working on that. The other one is to just load calcium indicators, and literally because this behavior, these cells only respond when you put a target, right? Otherwise, they're completely silent. Our hope is that we can actually image the brain with calcium indicators and see what actually flashes. And we think that what's going to happen is that with the rover flies, the central complex is going to light up because they're moving the head. They will normally move the head and the information routed through the central complex. Whereas we think that for the killer flies, what we're going to see is information going directly from the lobula all the way down, right? So I think that simple questions as such, how is the information being routed for now? And then by the time we get a handle on that, hopefully we should have the genetic tools to get going with it.
Any more questions?
There is no.
Can you raise those flies in the lab?
The killer flies we can, yeah, in big numbers. Whenever we don't get, things go wrong. But we are working on getting the other flies to rear them. We should be able to rear these guys as well. It's just a matter of how much effort you want to put into it, because they basically hunt down beetle larvae, and then they hook onto the little larvae and suck them dry. And so if it's going to take you a year to rear one of these guys, you may as well go out to the field and catch them. For the dragonflies, we catch the nymphs from the ponds, and then we rear them in the lab, for example, and then we can control the development and such. And there are some dragonfly species where it may work rearing them completely in the lab. So it's a lot of work just before you get into the neuroscience of things, yeah.
Do you see switches in the strategies that they're using? The reason I'm asking this is because this parallel navigation that you mentioned was also shown in other species, for example in bats. So Cindy Bowles many years ago showed that bats, when they attacked an insect, the insect is flying in a relatively straight line, the bat is using this exact parallel navigation. Of course, you're right that if they have to maneuver very fast, then there is inertia and things get complicated. But this made me think that maybe also the fly, for example, would change the gain as they approach the insect or like the prey or things like that. So have you looked at that? Or?
So when we look at erratic targets, it was actually easier to get the end correct because then you have all these turns, right, to test it. We think that it's quite consistent with these tiny animals because they don't really have to worry about anything else, right? Everything's kind of, when they turn, kind of they swim through the air anyway. For these guys are about this big, right? They're pretty big. And so we're excited about them because we can put things on them and record from them what they're flying. These guys, we can't figure out what they do because there's just so much variability, individual variability going on for exactly how they approach the prey, especially because they're beetle hunters. Once they get close to their prey, they circle it before catching it, right? So that breaks all the proportional navigation rules because they're doing something else. That now they use wind beat frequency to figure out that it was probably the right thing, and then once they get close, they really want to know it's the right thing. So I think what you're saying makes a lot of sense once the animal gets bigger and maybe the cognitive power gets, bigger as well. This is why we're excited to look at this. But for the small flies, I don't think it makes a difference.
Some other questions. So if you just compare like the gross anatomy, like using like NC82 antibody, is that like I don't know if that already exists, not for these different species, but is it like a particular increase in a particular region, like I don't know, like the VLP for example, or like the PVLP? Is there like a difference in the volume you compare across the species? I'm just wondering if like.
So we see, for example, the one that has the phobia, this guy here, that the eyes are wider, right? So the ventral nerve cord is exactly the same size, just slightly thicker, it's that bottleneck, right? But indeed, the lobula and the lobula plate are in completely different places because the retina has pushed everything away from it, right? And the phobia, the axons, let me show you, see if I can show you this picture quickly. So you can see, this is crazy, right? This is a normal fly eye, right? What this fly in here has done, let me see if I can show you more clearly. Oh no. so I cannot show you. Ah, there we go. Okay. So what this fly has done to fit the fovea into this eye, it's flattened the cornea at the front. It's almost completely flat. It's made huge lenses. It's made a very, very long focal length, and it's made very tiny photoreceptors, right? This is how you feed a fovea in the middle of a compound eye. And then you have to compensate for it and everything around here. So this is the size of the lenses and the size of the focal length that you will find on a fruit fly, for example, right? And so you can see the difference within the same eye, okay? And so the axons of these, when we look inside in this picture here in the middle, the axons of these photoreceptors here are huge. They're absolutely massive, right? And the amount of space that this area of the wall takes up here in the subsequent layers is huge as well. There's a lot more detail coming from this area here than there is from anything else. So definitely you get these effects where everything else matches basically the sensory input of these animals. So this is literally very, I'm the only person looking at predatory dipterans as far as I understand. And even in dragonflies, where there are many other labs that have been doing this forever, you know, there are recordings from different areas of the brain, but we don't even have an atlas of the brain. We don't even know what happens in the middle. So we have a lot of work to do on both these ones and the dragonflies. I'm just hoping that because these are dipterans, we're going to go much faster with this. Yeah.
Great. So thank you again. We'll be back at 10:45 with Jan Drakovic will be as well.